Hello, and welcome to our pre-lab on the integument. Integument, basically the skin, as well as the accessory structures of the skin, including the glands, the hair, and the nails. Now, if we take a look at the integument, again, as an organ system, what we're going to see are going to be two distinct layers, and then a third layer that we're going to include with this, just because it fits in here as opposed to someplace else. Within the integument, what we're going to have is an epidermis, and this is going to be indicated by 1 on this low magnification view of the area of the skin. So the epidermis is going to be primarily a maximally keratinized stratified squamous epithelium. And so again, we're going to have an epithelium, which is going to be found along the surface of the body. So we got the outside world here in this region. We're going to have the dermis, which is going to be deeper to the epidermis, and so we're going to have a connective tissue layer. And if we take a look at it at higher magnifications, we're going to see on a couple slides, we're going to have two distinct layers within the dermis. We're going to have a papillary layer of a dermis. This is going to be a loose connective tissue, essentially the lamina propria that we've talked about previously, kind of a smooth staining appearance, kind of immediately underlying our epidermis, underlying our epithelial lining. And then deeper to that is going to be the reticular layer of the dermis. And the reticular layer of the dermis is going to be a dense, irregular connective tissue. Kind of scattered among both the epidermis and the dermis are going to be those specializations we talked about, these modifications. In this case, we've got hair shafts, which are going to be present here. So you can see the hair shaft, you can see the hair follicle, the structure associated with this hair bulb over here at this point. Now, in most cases, these specializations associated with the skin are going to be structures that are going to be continuous with the epidermis. And so if we trace this hair follicle around, we can see how it's essentially continuous with the epithelial lining of the epidermis. Now, deeper to the dermis, over in here, we're going to be getting down into the hypodermis. And so what we're going to be looking at is going to be not actually part of the skin, but actually the subcutaneous fascia. It's going to be an adipose layer, which is going to be involved with anchoring the skin to the surrounding tissues. Now, there are a couple ways in which we can classify the skin. And so we can have thick skin or thin skin. And again, we've got a slightly different magnification here, but we've got the epidermis here running from about this point up. In this case, we got the epidermis down here to this bottom of the kind of purplish region. And what we're going to be looking at is in the thick skin, we're going to be looking at the skin that's located on the palms of the hands or the soles of the feet. We're going to have a very, very thick outer layer of the epidermis. There are going to be 5 layers of the epidermis we're going to talk about in the next slide. But the outermost layer, the stratum corneum, is going to be very, very thick in areas of the body that are subject to a lot of abrasion and, in essence, abuse. So the palms of the hands or the soles of the feet. In contrast, in thin skin, covering most of the rest of the body, we're going to have a very, very thin stratum corneum. And the layer, the stratum lucidum, is generally it's present, but it's not going to be able to be identified in light microscopy. And so if we take a look at this, again, thinking about the characteristics we talked about before, epidermis out here, dermis underlying it, so we've got the pinkish staining appearance here. Nice even staining for the papillary layer of the dermis. We got some white blood cells, some lymphocytes scattered in around here. And then deeper to the papillary layer of the dermis, we have the reticular layer of the dermis. So denser, coarser fiber bundles. Over here, again, papillary layer of the dermis, kind of smooth staining appearance, loose connective tissue. Deeper to that, we've got the reticular layer of the dermis, and we've got that coarse collagen bundles that are going to be present there and interwoven, giving us a lot of strength and integrity to our dermis. The second way of classifying skin is going to be either the presence of hair follicles, like we'd see in thin skin over here, good hair follicles, like we talked about a couple slides ago, versus hairless skin. Glabrous skin is going to be the skin that's found in like the palms of the hands and the soles of the feet. Now, when we take a look at the epidermis, take a look at the cells that are going to be present, what we're going to see, and this is a higher magnification view, so we don't see this outside space, which can be up here in this region, we can see the epidermis. And so it's going to extend down to the basement membrane, kind of about dashed along this line here. Loose connective tissue underlying it, the lamina propria. We're going to have 5 layers of the epidermis. We're going to have the stratum basale. This is that base level, that foundation level, 1 cell layer thick, where the cells are going to be dividing and pushing cells up into the epithelium. Stratum spinosum, relatively thick layer kind of spiny appearance for the spinosome aspect of it, because these cells are going to be connected together with a lot of desmosomes. They often contract a little bit or shrink down a little bit when we're processing them for histology. So normally they would be in close contact one another, pushing up against one another, but they often have a spiny appearance in a histological slide. Stratum granulosum, a layer of granular cells, because we can see the basophilic keratohyalin granules are going to be present there. The stratum lucidum, that fourth layer that should be at this point here, almost a translucent layer, it's not always apparent in hemotoxin and eosin stained specimens. But it's the area where we're going to have the establishment of the waterproof boundary. And then outside of that, we're going to have the stratum corneum, so non-nucleated cells, essentially dead cells, which are packed with keratin, so maximally keratinized with the keratin to the outside. Now, the predominant cell within the epidermis are going to be the keratinocytes. So the majority of these cells are going to be the keratinocytes. Got the stem cells down here within the stratum basale. They divide, push the cells up into the epidermis, and the cells are going to differentiate, essentially pack with keratin, die up here within the stratum corneum, and then be sloughed off anywhere from about 2 weeks to about 2 months later. So passing through the epithelium. We want to be able to continually provide new epithelial cells, new keratinocytes, so these cells are going to continue to divide throughout your lifetime. Melanocytes are going to be paler cells, which are found within the stratum basale. Again, these cells are going to be involved with producing melanin in those melanosomes, but they're not going to accumulate it. So their cytoplasm is going to be relatively pale, but they're going to inject, in essence, the melanosomes and melanin into the neighboring keratinocytes so that they have the ability to protect their nuclei from ultraviolet radiation and the harmful effects of free radicals. We go down from the epidermis into the dermis, again, because the keratinocytes and the melanosomes are going to be the predominant cells you can identify with hemotoxin eosin. We get down into the dermis, and so we're going to have the papillary layer of the dermis, and this is going to be the area which is going to be underlying those epidermal ridges, but it's going to be a loose connective tissue. So we're going to have some fibroblasts, lots of nucleated cells. These nucleated cells are primarily going to be lymphocytes. So migrating white blood cells are going to be going through the region to respond to potential pathogens that may get through the skin, and you want to essentially identify them and mount an immune response before there's a potential for them becoming established and causing an infection. So relatively smooth staining, lots of pink staining because of the thin collagen that's present here within the lamina propria, this loose connective tissue. More coarser staining over here in 2, where we've got the reticular layer of the dermis. Deeper, dense, irregular connective tissue, thick collagen fiber bundles through here, much less cellular. So the majority of the cells that are going to be present here are either going to be associated with larger blood vessels or lots of fibroblasts involved with maintaining the collagen or maintaining the connective tissue within the reticular layer of the dermis. Below that, we're going to have the hypodermis. Again, keep in mind the hypodermis isn't actually part of the skin, but we're covering here because it's going to be loose connective tissue and a lot of adipose tissue, white fat, which is going to be underlying the skin and attaching it to the surrounding areas. We've got a couple ducts over here associated with some sweat glands. We've got a very nice Pacinian corpuscle over here at this point, which we can also identify, at least in this slide. Now, taking a look at some of the skin specializations, we get the hair follicle. Again, most dominant structure in that is going to be the hair bulb. The hair bulb is going to look very similar to the epidermis, but it's going to have some modifications. It's going to have a dermal papillae underlying it, and then we're going to have cells which are going to be able to divide epidermal cells. They're going to be able to divide here and push the cells up. They're going to develop into the hair shaft. Now, if we trace this around, we're going to be able to see, the glassy membrane is going to continuous up with the basement membrane underneath the epidermis. These cells are going to be lining the spaces. Part of the hair follicle are going to be continuous with the epidermal cells of these stratified, maximally keratinized stratified squamous cells of the epidermis. During the anagen phase of the hair follicle, we're going to see the production of very nice hair shafts, again with the medulla that's going to be moderately keratinized, and the very thick cortex and cuticle where it's going to be heavily keratinized to perform the shafts of the hair. Now, associated with these hair follicles are going to be the sebaceous glands. Again, the sebaceous gland is going to be an exocrine gland involved with a holocrine mode of secretion. So these cells are going to essentially be the entire secretory product. And so you see regular looking cells down here at the bottom, regular nuclei. These cells divide, push themselves up into the gland-like structure, and as they pass through this gland-like structure, they're essentially going to become very differentiated, in essence, to become the secretory product. And so they're accumulating sebum, the product that's going to be coating the skin, lubricating it, having an antibacterial effect. But you take a look at the nuclei, and you're seeing the nuclei kind of condense down. They become pycnotic, which means that they're going to be condensed, they're going to look fragmented, and ultimately they're going to be lost. So that by the time the cells are up here, kind of closest to the hair shaft, to the hair follicle, they're going to be, in essence, dead cells. They're going to be dumped onto it to coat the hair shaft itself. Eccrine sweat glands are going to be simple coil glands. They're going to be characterized by lining of a simple cuboidal or columnar epithelium, normally a cuboidal epithelium, relatively small lumen in relationship to the thickness of the wall structure. And you can find these in most areas of the skin except for the lips and portions of the external genitalia. Again, characterized by a nice duct system, secretory cells associated with it, but relatively small lumen in relationship to the thickness of the wall structure. So it's going to be a simple, normally cuboidal epithelial duct system within the body are going to be the eccrine sweat glands. Relatively small lumen in relationship to the thickness of the wall structure. In contrast, the apocrine sweat glands are still going to be lined by a simple cuboidal epithelium, but they're going to have a very, very wide lumen in relationship to the thickness of the epithelial lining. The apocrine sweat glands are going to be found in areas like the axilla, areola, nipple, perianal regions, and external genitalia. And so you may be able to find these in some of the histological specimens, but they're going to be a little bit rarer than the eccrine sweat glands that are going to be more common within slides of the skin. And then the final specializations we're going to talk about are encapsulated sensory receptors. And so the first is going to be the Meissner's corpuscle. Now the Meissner's corpuscle is going to be found in the dermal ridges, in the dermal papillae. So if you take a look at them, look in the loose connective tissue, kind of immediately underlying the epidermis up here to the top. And so when we have one of these dermal papillae kind of extending up, what we're going to have is kind of a zigzag pattern of modified Schwann cells, and this is going to be a specialized touch receptor. And so you can find Meissner's corpuscles very prominently in the skin of the lips and the fingertips. The second type of encapsulated sensory receptor that you're going to be able to see within these histological specimens and be able to identify them in H&E light microscopy specimens are going to be the Pacinian corpuscles. Now, the Pacinian corpuscles can be found deeper within the dermis or into the hypodermis itself. And that's the example we've got in the slide here. So you're going to see like an almost like an onion-shaped structure. The Pacinian corpuscle is going to have alternating layers of fibroblasts and fluid-filled spaces, some thin collagen fibers holding the thing together. And it's going to appear like a sliced onion. Now, these things are going to be able, the Pacinian corpuscles are going to be able to respond to either vibration or pressure. So pressure that's deep enough to kind of deform this Pacinian corpuscle, deep enough to push down all the way through the dermis, the epidermis and the dermis, all the way through the skin and push on it, or vibration of the structure itself. And so either deformation pushing on it or vibration is going to cause the fluid in these layers to move in relationship to one another. And that's going to trigger the Pacinian corpuscle to send an impulse to indicate that we either have pressure or vibration going on within this region of the body. And this finishes up our pre-lab activity associated with the integument with the skin. As always, if you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thank you.